before I start, let me ask you one question. What are the basic tissues of our body? How many types of tissues are there in our body? 4 types are there. What are they? First, epithelial tissue, then connective tissue, muscular tissue, and nervous tissue. Okay, these are the 4 basic tissues of our body. Among them, I hope you have studied about the epithelial tissue, right? Epithelial tissue is done. Have you studied about muscular tissue? What we'll do, first we'll discuss about the connective tissue. Okay, so it is also a type of tissue. What is connective tissue? Then how the connective tissue is formed, what are the components of connective tissue? Okay, then classification of connective tissue. Okay, then I'll show you some slides, microscopic slide, where we'll see how the connective tissue look like. Okay, followed by cartilage, because cartilage is also a part of connective tissue. It is a specialized connective tissue. Okay, 3 specialized connective tissues are there. Okay, so among them blood, bone, and cartilage. So among them cartilage is also a type of connective tissue. So today I will try to finish the cartilage chapter also. Now, don't think that this particular demonstration class will be like lecture. No, in this class we will focus mainly on the practical aspect. Now in histology, the most important thing is you have to identify the slides. That is the most important thing. Okay. Now during the time of exam, this particular slides will be kept. Okay, and it will be covered like this. Now if it is covered, you can't see. So then how you will identify? Now if I remove it, you can identify it's written over here. Now suppose if I cover it like this and if I put it under microscope, how will I identify? With the help of the identifying features. That means you have to know about the tissue, then only you can identify. Okay, so during the time of exam, this kind of slides will be placed under the microscope. And for identification and writing the identifying features, 2 minutes. You will be getting only 2 minutes. Within that you have to identify, and at least you have to write 2 to 3 identifying features. Then you have to go to the next slide. That's become a bit challenging. So for that, what we have to do, everyone, please make it a habit. Whenever you are coming for this histo demonstration class, after demonstration, okay, whatever slides are available, go to the histo lab. Everyone, go to the histo lab, take the slide, try to focus it, and then try to identify. I will also be there, but you have to practice. Okay. So once you will practice, then you will come to know that which particular slide it is. Then what you should do, what you have to do. Now suppose you have focused, you have identified the identifying features, then with the help of your phone, just take a photo of that particular slide. Okay. And then save it as the name given in the slide, and it will be better if you make a separate folder, so that before the exam you will be able to see all the slides. Okay, and if you see if you revise for more than 3 or 4 times of one single slide, then something will be there in your mind, and during the time of exam that will help you to identify the slides. Practical exam, final practical exam. Okay, so first we'll discuss about the connective tissue, and then I will try to complete the cartilage part also. Okay, and then we'll go to the lab. Okay, so 10 students will form one group, we'll go to the lab, we'll see the slide. After that, next 10 students like this will rotate. Okay. So what is this connective tissue? So connective tissue is a type of tissue which connects, separates, supports all other tissues of the body, and it also fills the interstices between the more specialized elements to serve them and hold them together. That is the definition of connective tissue. Okay, that means suppose different organs are there, different structures are there, or different tissues are there, to hold them together, to support them, to separate them from each other, the connective tissue is present. Okay, that is the connective tissue. Now, if you see the connective tissue, if you see the difference between the epithelial tissue and connective tissue, in case of epithelial tissue, we have seen that cells are placed very closely. If you take the example of cuboidal epithelium or columnar epithelium, they are placed very closely. In between the cells, gap is not there. But here in case of connective tissue, the cells are distributed. Wide space is there in between the cell. And along with that, we are also having the fibers. Different types of fibers are present. And all those constituents are held together by a gel-like substance that is called extracellular matrix. Okay, which is holding all the ingredients together. Okay. Now, in case of connective tissue, differ from other type of tissue is that the cells are loosely arranged rather than tightly packed within the extracellular matrix. Okay, so cells are loosely arranged. Let me show you a diagram. Yeah, so here you can see the diagram of connective tissue. Okay, this is a schematic diagram of connective tissue where you can see all the parts of the connective tissue. Can you see the cells? Okay, these are the cells of the connective tissue. Now, can you see in between the cells huge gap is there? Not like epithelial tissue where cells are very closely placed. Okay, gaps are there. Now, in between them, can you see different types of fibers are present? See, this is one type of fiber, this is one type of fiber, this is another type of fibers. Okay, different types of fibers are there. And all these are supported together or holded together by this extracellular matrix. Okay. Now, this particular area which you can see, this particular area, this is nothing but the extracellular matrix which is holding all those things. So, this is the constituent of the connective tissue. Now, the structure of connective tissue, if you want to study the structure of connective tissue, there are three components of the connective tissue. What are these three components? What are the three components? Cells, fibers and ground substance. As you can see, cells, fibers and ground substance. These are the components of connective tissue. Okay. Now, here I will give you a shortcut how to remember connective tissue. What is the first alphabet? C. Number? Third. So, how many components? 3. Okay. So, 3 for 3. So, like this you should study so that you will never forget. Okay. So, connective tissue, how many components? 3 components are there. Okay. Cells, fibers and ground substance. Now, so the cells are there, ground substance is there and fibers which I have shown you in a diagram. Okay. Now, we will be discussing about each and every type of cells. Okay. We will be discussing about the fibers and then we will be discussing about the ground substance. In brief, not in detail. Okay. Now, please keep that in mind. 2 types of cells are there. In case of connective tissue, 2 types of cells are there. Okay. What are they? Fixed cells and free cells. Fixed cells and free cells. Free cells are also called as wandering cells. Now, what is the meaning of this? Meaning is free cells are not the part of connective tissue. They are coming from somewhere else to the connective tissue. Okay. Now, can you see the list of the cells, free cells? Have you heard about these cells? Where you have heard? Yes? Yes. Okay. That means you have studied all those things. So, these are the cells of bloodstream. From bloodstream, they are entering the connective tissue. The function is same. Okay, though they are not the part and parcel of connective tissue, that's why they are called as fixed free cells or wandering cells. Okay, next here you can see the fixed cells. Okay, fixed cells means permanent cell. They are present only in the connective tissue. Okay, that means in the different types of connective tissues. So you can see fibroblasts and fibrocytes are there. Okay, so fibrocytes are not same. It is different. It is having some difference, we will see. Next, you can see macrophages are there. I hope you have heard about macrophages, also called as histocytes. Function is phagocytosis. Next, mast cells are there. I hope you have heard about the mast cells also. Okay. A precursor of histamine. Next, adipocytes are also there. Okay. Now, adipocytes are also called as fat cells which can store fat droplet. Okay. An aggregation of the adipocytes forms the adipose tissue. Next, pigment cells are there, also called as chromatophores. They are more in case of skin, then iris of the eyeball. and then reticular cells which are present in reticular connective tissue. Okay, so these are the different types of cells, fixed cells which are present in the connective tissue. Now let me show you a diagram of the different types of cells, okay, which are present in the connective tissue. Now here you can see the cells of the connective tissue. Okay, so first you can see this is the fibroblast. Just see the appearance of the fibroblast. Then this is the plasma cell, this is the adipose cell, lymphocytes, then macrophages, okay, fibrocytes, eosinophil, neutrophil, okay, this is the mast cell. So this is the appearance of the cells, okay, both free and the fixed cells which are present in where? Connective tissue. Okay. So now we'll be discussing about the fixed cells, because free cells already you have studied in physiology. I know that functions are same. Okay, so without wasting time, we'll directly go into the fixed cells. Okay, so in fixed cells, which is the most abundant cells? Anyone? Any guess? In fixed cells, okay, I have shown you the list. Among them, which is the most abundant cells? Fixed. Yes, very good. This is the fibroblast, this one. Okay, this is the most abundant cells. Okay, fibroblast. And they are having very significant role in the connective tissue. So fibroblasts, as I said, present in the connective tissue. Now, this particular fibroblast, they produce collagen fibers, different types of collagen fibers. They produce the proteoglycans, fibronectin, laminin, glycosaminoglycans, metalloproteinases, prostaglandins, etc. These are the parts which are secreted, or you can say produced by this fibroblast. Okay, and these particular chemical compounds, whatever I have mentioned over here, they are present in the ground substance. That means you can say that this particular fibroblast is not only forming the fibers, they are also laying down the ground substance or the matrix for the connective tissue. That's why fibroblast cells are very important.
Sir,
Yes. About the fibroblast. Fibroblast, as I said, these are the most abundant type of cells in the connective tissue. Now, the main function of the fibroblast is to produce collagen fibers, different types of collagen fibers. Along with that, it also secretes these proteoglycans, fibronectin, laminin, glycosaminoglycans, etc. Now, these particular things, chemical compounds, they are present in the ground substance. So, when you will study the ground substance, at that time, you will see what are the ingredients present in the ground substance. Okay. Next. Now, fibroblasts are stem cell origin, means they originated from the stem cell. If you see the shape, elongated, spindle shaped or stellate shaped, that is the shape of the fibroblast. Okay. Along with that, they are having cytoplasmic projections, which I have shown you just now. Now, within the cytoplasm, abundance rough RER is there, and along with that, large Golgi apparatus. Okay. Now, this particular fibroblast, when they are associated with the reticular fibers, then they are called as reticular cells. Okay. So, we'll be studying about the fibers also. So, reticular fibers are there. So, when it is associated with reticular fibers, then these are called as reticular cells. Next, after that, fibrocytes are there. So, fibrocytes, these are inactive form of fibroblast. Okay, or also you can say that immature cells. Now, they are smaller and thinner and morphologically different from the fibroblast. Okay, that means the appearance is also different. They are immature cell. They will mature and then they will become the fibroblast. Okay. Next, macrophages are there. So already you know what is the function of the macrophages. And the other name is histocyte. The other name of macrophages are also called as histocytes. Now you know what is the function. That is the phagocytosis. They will eat up the debris or the part of the bacteria which is invading the connective tissue. Next, mast cells are there, which are round oval in shape. They are also called as histaminocytes. Now, the nucleus, in case of mast cells, they are centrally placed. Irregular microvilli are present on the surface, and the most important feature is numerous granules present inside the cytoplasm. That is one of the striking feature of the mast cell. Okay. Next, fat cells are there, also called as lipocytes. What is the shape of the fat cells? Anyone? Ring-like. Okay. If you see the shape of the fat cells, they are ring-like. So inside the cell, what is present? Fat droplet is present. And that fat droplet is pressing or compressing the cellular organelles towards the periphery. Okay, so that nucleus part which is there, it forms a small bulging. That's why it appears as ring. Okay, ring-like appearance, or also called as signet ring-like appearance. Lastly, the pigment cell, they are also stellate shaped, easily distinguished as they contain brown pigment that is called as melanin. Now, the most abundant connective tissue of skin, choroid and iris of the eyeball. So, these are the areas where this particular pigment cells are present mostly. Okay. Okay. Next, so 3 components are there. First component was cells. Second component, fibers, and then ground substance. Okay, so how many types of fibers are there? Again, 3 types of fibers are there. What are they? Collagen fibers, elastic fibers and reticular fibers. These are the 3 different types of fibers. Now, before we start with the collagen fibers, have you ever seen a electron microscope image? Till now, whatever histological images you have seen, these are of compound microscope, this type of light compound microscope. Okay, now the images which are produced by this type of microscope and the images produced by electron microscope, huge differences are there. Okay, fine detail can be seen. Even a small cellular structure, what is the shape of the cell, what are the processes, even microvilli, all those things can be seen under electron microscope. All those things you can't see under a normal light microscope. Okay, so I am having an image of collagen fibers under electron microscope. Okay, now here you can see the collagen fiber. This is the collagen fiber. This is the collagen fiber. Now see the appearance. Okay, and this is the electron microscope image of collagen fibers. See the difference. So in fibers, what we have to discuss, what we have to study, we have to study about the characteristics of the fibers. Okay, means where these fibers are present, and mainly what are the function of these particular fibers. So we'll start with the collagen fibers. Now please keep that in mind, the collagen fibers always run in bundles. The collagen fibers, whatever it is there in our body, any part of our body, collagen fibers always they run in bundles. Means not a single fiber. So many fibers, they will make a bundle and those bundles are running. Okay. Next point. The bundles which are formed, they may be straight or they may be wavy. The bundles which are there, it may be straight or it may be wavy depending on the force which is applied on it. Okay. Next point. The individual fibers which are present inside the bundle, they are always parallel to each other. Okay, so these 3 points you should remember about the collagen fibers. First, the fibers together, they will form a bundle. Now the bundles, they may run straight or they may be wavy depending on the force applied on it. Next point is the individual fibers, whatever are there inside the bundle, okay, they will run always parallel to each other. Okay, now the bundles which are there, they can branch anastomose with each other, but the individual fibers, they never branch anastomose, they will always run parallel to each other. Okay, these are the important properties of the collagen fibers. Next, the bundles are made up of collection of individual collagen fibers having diameter of 1 to 12 micrometer. Then bundles often branch or anastomose with adjacent bundles, but the individual fibers do not branch. Okay. Next, with electron microscope, each collagen fiber seen to be made up of fibrils, that is having diameter of 20 to 200 nanometer. Now what is the meaning of this? For that I have shown you the electron microscope image. Okay, so with the help of compound microscope you can see the bundles. Beyond that you can't see anything. Now if you want to see beyond that, you need electron microscope. So with the help of electron microscope, what you can see? You can see that the collagen fiber, individual collagen fiber are made up of fibrils. Okay, and this fibril is having diameter of 20 to 200 nanometer. Next, each fibril consists of again microfibrils having 3 to 5 nanometer in diameter. So see, we are going more deeper. Okay. So first, one particular fiber, you have taken out one fiber, then if you see that particular fiber under electron microscope, what you will see? You will see that it is made up of fibrils. Now if you magnify it more, then you will be able to see that this particular fibril, again it is made up of microfibrils. Okay, and the diameter is 3.5, approximately 3.5 nanometer in diameter. Next, the collagen is made up of molecules that is called as tropocollagen, very important. Now these microfibrils of the collagen are chain of tropocollagen molecules. Okay, tropocollagen is a protein. So how they are formed? These particular microfibrils, the microfibrils of the collagen are chain of tropocollagen molecules. Now each molecule of tropocollagen is having length of 300 nanometer, the length, and the molecules of tropocollagen are arranged in regular overlapping pattern. Okay, so the chain is regular and each chain overlaps another one. That's why we are getting a striation or cross-striated appearance of the fibers. And this cross-striated appearance of the fibers can be seen under electron microscope only. Okay, so these are also some unique features of the collagen fibers. Next, there are different types of collagen fibers in our body. Can you guess how many types of collagen fibers will be there? Any idea? It's more than 20. More than 20 types of collagen fibers are there in our body. Okay, now among them, in your histology book I have seen up to type 5 it is given. Beyond that it's not there. Okay, researches are still going on, but different types of collagen fibers are also there. Now previously I have shown you the electron microscope image. Now this is the normal compound microscope image. So see, these are the bundles of collagen fiber. These are also the bundles of collagen fiber. Now this is LS, this is TS. I hope you know what is LSTS, right? So just see how the collagen fibers are arranged. You can see they are running in bundles. Okay, you can see they are running in bundles. So only you can see the bundle. Beyond that you can't see anything. Same thing is present over here also. You can see these are the bundles of the collagen fibers. Okay, one LS and another one is TS. Beyond that, if you want, you have to use electron microscope in order to see the fibrils, microfibrils, those tropocollagen, all those things. For that you need electron microscope and the cross striations. Now, so if I ask you what is the example of collagen fibers where they are present? Best example, ligament. Okay, then the capsule of the synovial joint. Okay, next type of fibers are elastic fibers. Okay, so here you can see the elastic fibers. Here also you can see the elastic fiber. Can anyone differentiate between these two diagrams? Can you differentiate between these two diagrams? Mm-hmm. Yes? No. See the color, whether is it same? Color is different. Why?
Staining.
Yes. Staining is different. Okay. So I hope you know that whenever we are preparing a slide, in order to see under microscope, we have to apply stain. If we don't put stain, we can't see anything. Okay, we have to put stain. Different types of stains are there. Most of the slide, whatever you see in the histo lab, which stain is used? H&E stain. Okay, now there are other stains also, and all the tissues of our body will not take the H&E stain. Different type of stains are there, which are used to stain different types of tissues. Okay, so that's why I have shown you this too. Now, which stain can be used? We'll be discussing. I'll show you which different types of stains are used in case of reticular fibers, in case of elastic fibers. Okay, I'll show you. Now what are the properties of the elastic fibers? Now in case of elastic fibers, they are very less as compared to the collagen fibers in our body. They always run singly, not in bundles. Okay, previously we have seen collagen fibers, they are running in bundles, but here single fiber will run. They will branch and anastomose with each other. Okay, as you can see, they will branch anastomose with each other. Now elastic fibers are thinner to those of the collagen fibers. That means the diameter is less. Okay. Now in some situation elastic fibers are thick. Okay, there are some areas where it is thick. For example, ligamentum flava, where it is thick. Then there are some areas where it forms the fenestrated membranes. For example, large arteries, walls of the large arteries. Now have you heard about this one, ligamentum flava? Anyone? No, because you have not studied the vertebral column. You have not studied the vertebrae. Okay, you have seen the skeleton, right? Everyone, in skeleton you have seen the vertebral column. Then a vertebral column, it is made up of vertebrae. So in between the vertebrae, different ligaments are there. Among them, one is the ligamentum flava. Okay, so in that case, the fibers are thick. Okay, next, fenestrated membranes means the membrane which is formed having small microscopic pores, small pores, tiny pores are there. Those are called as fenestrated membranes. Okay. Next, again, so this is the electron microscope image, not direct electron microscope image, a schematic diagram of a electron microscope image of the elastic fibers, where you can see it is made up of central amorphous core and layer of fibrils. So this is the central amorphous core, which is made up of a protein that is called as elastin. Central core, it is made up of a protein that is called as elastin. Okay. Around that, can you see these dot things? These are nothing but the microfilaments. Now these microfilaments are made up of fibrillin, which is nothing but a glycoprotein. Okay, so this is the structure of elastic fibers. So normally the elastic fibers which you will see under compound microscope is they branch anastomose with each other, single fiber. That much you can see. Beyond that you can't see. Okay, and all those things I will show you today under microscope. Next, these are the reticular fibers. Okay, now just now I have told you different types of collagen fibers are there. More than 20 types of collagen fibers are there. Okay. Different types of collagen fibers are there in our body. Among them, type 3 collagen fiber is nothing but the reticular fibers. Okay, type 3 collagen fibers are reticular fibers. Okay, that's why I told you that different varieties of collagen fibers are present in our body. Okay, now each and every variety, some properties are different, arrangement of the fibers are different, not same. That's why type 1, type 2, type 3. Now reticular fibers, as I said, the variety of type 3 collagen fibers. Okay. Now they also shows the periodic striations. They are much finer, uneven in thickness. Now they also branch anastomosis with each other. But the difference is, in case of these reticular fibers, whenever they branch anastomose, they are forming a network-like structure, and that is also called as reticulum. Okay. Elastic fibers, they also branch anastomose, but here what is the difference? Whenever they branch anastomose, they are forming a network-like structure that is called as a reticulum. That is the difference. So as you can see, they form a network or reticulum by branching and anastomosing with each other, and they do not run in bundles. Okay, the reticular fibers also, they do not run in bundles. Now it can be easily distinguished from type 1 collagen fibers as they are stained brown. Okay, why? Because you can't stain them with the help of H&E stain. Okay, जो reticular fibers होता है, you can't stain them with the help of H&E stain. For that, a special type of stain are used, and they are made up of silver salt. They are made up of silver salt, silver nitrate, AgNO3. That's why. See, they are having affinity for silver salts. That's why they are also called as argentophil fibers. Okay, that's why they are also called as argentophil fibers. Now, where these reticular fibers are present, now these reticular fibers provide supporting network in many situations, supporting framework. For example, in case of spleen, lymph nodes, bone marrow, most of the glands, liver, kidney, etc. So, in those organs, the framework is formed by the reticular fibers. Okay, that means these are the areas where reticular fibers are present. Okay. From here the short questions are asked. So always whenever you are studying about any epithelial tissue, any connective tissue, cartilage, at that time what is the location? For example, hyaline cartilage is there. What is the location? Basic question. Okay, that you have to by heart, that you have to know, because all those questions will be asked in the practical exam. Whatever we have discussed, this is the gist of the story. Okay. So what is written over here? First, you can see collagen fibers, elastic fibers and reticular fibers. Okay? Then you can see the color, what is the appearance, then number, which is more, which is less. Then stain appearance, means whenever you are staining, okay, at that time what is the appearance. Now as I said, reticular fibers you can't stain with the help of H&E. For that, a special stain is used, that is the AgNO3 or silver nitrate stains are used. After that, what will be the appearance? It will be black. That's why you can see blackish appearances are there. Okay. Next, what are the sites, as you can see, to provide strength? For example, tendon, ligament. This to provide elasticity, ligamentum, nuclei, vocal cords, inside the lung also. And this to provide support, spleen, liver, lymph nodes, kidney, basement membrane, etc. These are the areas where these reticular tissues are found. Okay. So that is all about the fibers. After that, what is the last one? Ground substance. Okay, so how the ground substance are formed? So ground substance, it is transparent material, a viscous solution, highly hydrated thin gel. Okay, principal constituents are large carbohydrate molecules. Along with that, complex protein and carbs are also present, and these are called as glycosaminoglycans. Okay, this is the constituent of the ground substance. Now what is the function? Now it acts as gel-like matrix which fills the spaces between the cells and fibers, provides support, allows nutrient exchange. Very important. Fibers are there, cells are there. How will they get the nutrition? With the help of the ground substance only, by diffusion. Okay, because the blood vessels are present over there. Next, it also helps in arresting the compression force, plays a major role in cell migration and along with proliferation and tissue development when there is any damage. So these are some important functions of the ground substance. Okay. Now this is the functions of the connective tissue. As you can see, enclosing and separating the organs. After that, connecting tissues to other, for example, ligaments and tendons, supporting and moving, mostly present in the joints. Okay, the cartilage. Just now we'll study about the cartilage. Okay, we'll be studying about the cartilage. At that time, we'll come to know how it is supporting. So whenever you are doing any movement, okay, suppose you are eating, at that time you are doing movement. Whenever you are walking, at that time also you are doing movement. So most of the movements, where they are taking place? In the synovial joints. You have studied about synovial joint, right? So those movements are taking place in the synovial joint. So whenever those movements are taking place in the synovial joint, okay, by definition you know that in synovial joint 2 or more than 2 bones will be there and they will be united together. Now when they are united together, a small gap is there and that is called the joint cavity, and the articular surfaces which is forming the joint is covered by a cartilage, covered by a connective tissue, and that is called as cartilage. If the cartilage is damaged, at that time there will be friction between the bones. Patients start suffering from pain whenever they are doing certain movements. Okay, so that's why this cartilage is also a connective tissue. So that's why supporting and moving. Then storing adipose tissue. Adipose tissue is to store fat. That is also important function. Then cushioning and insulating, again adipose tissue. Transport, protection by the blood and protection by the cranium and sternum. Okay, so bone is also a type of connective tissue. I hope you know that. Specialized connective tissue, but it is a connective tissue. Okay, which is protecting. For example, sternum, very important. Why it is important? Any idea? Sternum. What are the? Yes, yes. No. See, sternum, three parts are there of the sternum, right? Manubrium, body and xiphoid process. Okay. So the strongest part of the sternum is the manubrium. A manubrium is protecting the great vessels of our body, not the heart. Heart यहाँ पे नहीं रहता है, इसके नीचे रहता है. Okay, protecting the great vessels of our body. So suppose whenever any accident takes place or anything happens, this particular sternum will protect the great vessels of our body. Okay, so this is the protection. Okay, next the classification of the connective tissue. Okay, so two types of connective tissues are there: connective tissue proper and specialized connective tissue. Connective tissue proper and specialized connective tissue. So, what is the difference between this? Any idea? What are the differences between these two? See, this is the connective tissue proper, or also called as generalized connective tissue, and these are specialized connective tissue. Okay, so under generalized you can see loose areolar, dense regular, dense irregular, elastic adipose and reticular connective tissue. Okay, and then under specialized bone, cartilage and blood. Now my question is, what do you think, what will be the differences between them? Why they are categorized separately? Why they are not kept together, though they are the part of connective tissue? No. Cells are present everywhere. Name of the cells are different, but cells are present everywhere. Here also cells are present, here also cells are present. Huh? Not exactly. The difference lies on the modification of ground substance. Modification of the ground substance. Okay, based on that the classification is done. Okay, so first we will see the loose areolar connective tissue. Okay, so loose areolar connective tissue, if you take the loose areolar connective tissue, it is having all the components of the connective tissue. You can see mainly the fibroblast, and you can see some macrophages. Along with that, you can see the collagen fibers also. Along with that, white elastic fibers are also present in the loose areolar connective tissue. Okay. Next, the material of the connective tissue gives loose consistency similar to the cotton ball when it is pulled apart. If you see the loose areolar connective tissue, okay, the consistency of the loose areolar connective tissue is similar to the cotton ball when it is pulled apart. For that, it is used around the blood vessels mainly to provide cushioning effect. Now, I hope you have seen that whenever you order something from those Flipkart and all, when the parcel is delivered, when you open it, what do you see? They used to pack it properly with the help of thermocol and with the help of other sponge-like material. Okay, why? Because that particular structure should not, object should not get any damage. So similarly, these loose areolar connective tissue, they are present around the blood vessels mainly. Okay, as you can see, blood vessels helping them to keep the vessels in place. They are also found around and between the most of the body organs. Okay, that is nothing but the cushioning effect is provided by the loose areolar connective tissue. Okay, now here is the ideal diagram of loose areolar connective tissue. Okay, this is not a histological diagram, this is a schematic diagram. Okay, next adipose tissue. The adipose tissue already we have discussed, right? You know adipocytes are there. Okay, this will store the fat droplet is present inside it. Okay, the cells are ring-like, signet ring appearance. Now where they are present? They are present under the skin, kidney, behind the kidney. Okay, you can see a huge amount of adipose tissue, and on the heart. I hope mediastinum is going on. Have you seen the heart? Yes. Okay, so next time when you will go to the dissection hall, please see it carefully. On the anterior or the sternocostal surface of the heart, you can see a yellow color coating is there. Okay, in some cases it is much, in some cases it is less, but still a small amount of yellow color substance you can see it will be there. So that is nothing but the adipose tissue. Okay, if it is more, then the person is having some trouble. Person may have some problem. Okay, but deposition is there. You can check. So what is this? This is the adipose tissue. This is a histological image of adipose tissue. This is how the adipose tissue appears. Now I have told you that what is the shape of the cells, adipocytes, ring-like. Okay, to make you understand, it is written that it is ring-like. This is the original image. Can you see? Is it ring-like? No. So actually, it is not ring-like. But to make you understand, it is written that it is ring-like. In actual sense, if you take any of the slide under microscope, you won't be see that exactly it is ring-like. Okay, for a reference, it is being taken that yes, it is ring-like. Now, why these particular parts are empty? Anyone? Okay, so you can see these are the adipocytes. These are the adipocytes. These are the adipocytes. Okay. Now, you can see the cell membrane. You can see the nucleus also, right, which is pushed towards the periphery, and the middle of the cell, can you see the middle of the cell? It is totally empty. Okay, why? Because see, first of all, fat droplet is there. Okay, now when we are preparing the slide, when we are preparing the tissue, at that time various steps are there. Among them, one step is called xylene wash. You have to wash it with xylene. When that step is done, at that time the fat droplet is washed away. So if it is washed away, nothing is there. So whenever you are giving the stain, if nothing is there, so it can't take the stain. That's why it is appearing empty. Okay. Next, dense regular, dense irregular connective tissue. Okay. Now, what is the difference between dense regular and dense irregular? In both the cases, bundles of collagen fibers are there. Okay. As you can see, यहाँ पे भी देखो bundles of collagen fibers are there. यहाँ पे भी bundles of collagen fibers are there. But the difference is here, they are regularly arranged. Okay, as you can see, they are parallel bundles of collagen fibers. They are regularly arranged. Now here they are irregularly arranged. Mainly they are interwoven. Okay, that is the difference between the dense regular and dense irregular. Now this particular found in the portion of the skin, they are found in tendons, ligaments. These are also found in the capsules of the synovial joint. Okay, this is dense regular and dense irregular. Next is a reticular connective tissue. So it contains reticular fibers and reticular cells. So previously we have discussed, okay, that whenever the fibroblasts are present along with the reticular tissue, then it is called as reticular cells. Now location, it is present in the supporting framework of liver, spleen, lymph node, etc. Okay, always the reticular tissue are providing a supporting framework of that particular organ wherever they are situated. Okay. Next, their function binds together smooth muscle tissue, filters and removes the microbes in the lymph node. These are some important functions. So when you will study the lymph node, okay, lymph node is a part of lymphatic system. When the lymph is flowing through the lymphatic channel, at that time they has to pass through the lymph node. And lymph node is nothing but the filtration of the lymph which is passing through it. It will filter the lymph. Now, whenever the filtration is done, okay, that particular structure is made up of this reticular tissue. Okay. Next is the elastic connective tissue. Now, it is consisting of elastic fibers. Fibroblasts are present in between the fibers and yellowish in color because of the elastic fibers. Location, they are present in tissues, walls, elastic arteries, trachea, bronchial tubes, vocal cords, etc. These are the areas where this particular elastic connective tissues are present. So I hope you have observed that every time I am mentioning the location of that particular tissue. That is very important. And in the beginning I have told you. Okay. And then functions, as you can see, it allows stretching of various organs or various structures because elastic fibers are present. Okay. So that is all about the generalized connective tissue. Next one is the specialized connective tissue. Okay. In specialized connective tissue, as you can see, bone, cartilage and blood are there. Okay. Among them, what we will study today? The cartilage. Okay. Now, along with the cartilage, I will show you only one slide. I have seen only one slide of adipose tissue, okay, from the connective tissue, which I can show, and already I have shown you. Okay, so again what you have to do, you have to see it under microscope. So for that, we'll go to the lab. First I will focus it, I will show it to you, then you have to focus. I will take out the slide, you have to focus, and then you have to see, try to identify the cells. Okay, same thing we'll be doing for the cartilage also. Okay. Okay, so we'll start with the cartilage. So cartilage is a specialized connective tissue. Okay, and as I said, what is the difference between specialized connective tissue and generalized connective tissue? Modification in the ground substance. Okay, so what are the modifications present in the cartilage, especially in the ground substance, that you have to understand. Okay, so cartilage is a specialized connective tissue, provides rigidity along with elasticity. That is the speciality of the cartilage. Okay, it will provide rigidity, along with that it will also provide elasticity. Now it is present in the body where both rigidity and elasticity are required. Okay, and in cartilage the cells which are present, these are called as chondrocytes, and they are found in a gel-like matrix. Okay, now if you see the cartilage, they are much more older as compared to the bones. Do you know that? Because I think you have not studied about the development of the bones. Development of the bones studied. So how the bones develop? From a cartilaginous model, the ossification center arrives, and then the bone formation is taking place. Ossification is taking place, right? So what does it mean? That means that first cartilage is there, and then from cartilage we are getting the bone. So that means cartilages are more older as compared to the bones of our body. Okay. Now the cartilages are replaced by bone, and these are called as temporary cartilages. Okay, and there are some cartilages which are permanent throughout the life. Can anyone give me the example of this temporary cartilage? Do you know the growing part of a bone, long bone? What are they? Epiphysis, then epiphyseal plate, metaphysis and diaphysis. These are the parts of growing long bone. Now what is this epiphyseal plate?
The plate between the epiphysis and
Yeah, it is made up of what?
it's hyaline cartilage.
Yes, it is a cartilage. Okay, so as the age advances, what happens? That is replaced by bone. Okay, so that means these are temporary cartilages. That's why I am asking you to give an example. Permanent one, permanent cartilage, the best example is trachea. Whenever you will study the trachea, at that time you will come to know that C-shaped tracheal rings are there. Those are made up of hyaline cartilage. They are permanent, they don't ossify. Okay. Okay. General features of the cartilage. So what are the general features of the cartilage? As you can see, the cartilage supports the regions of the body that requires flexibility along with rigidity. Okay. Next, it is not having blood supply and it is non-nervous structure. What is the meaning of this? No blood supply, no nerve supply. Okay. For that, there is a problem. What is the problem? Poor regeneration capacity. That means if there is any damage to the cartilage, okay, the healing is very slow. As a result, it is replaced by fibrous tissue because it don't have direct blood supply and nerve supply. Okay. Next, consists of all three components of the connective tissue, usually surrounded by perichondrium. Perichondrium is the outer covering of the cartilage. What is periosteum? Outer covering of bone. Similarly, the cartilage is having outer covering that is called as perichondrium. Okay. Now there are some areas where perichondrium are absent. For example, articular cartilage and fibrocartilage. Okay, these are the areas where perichondrium is absent. Now the perichondrium which is there, generally two layers are there in the perichondrium. Okay, fibrous layer and cellular layer. So in fibrous layer some blood vessels are there. So the cartilage is getting nutrition by diffusion from that blood vessels which are present in the perichondrium. That is the only source of the nutrition for cartilage. Okay. Next, what are the functions of cartilage? So due to the extracellular matrix, It allows the tissue to bear the mechanical stress on it. Then in respiratory tracts, ear, nose, cartilage forms the framework of the supporting soft tissue, provides shock absorbing and sliding region, especially in the joints, which facilitate the movements. Okay, so these are some important functions of the cartilage. Next, the components of the cartilage. So we know that cartilage is a specialized connective tissue. Okay, that means all the components will be there. So it will be having cells, it will be having ground substance, and it will be having fibers also. Okay, so in cells only two types of cells are there, chondroblast and chondrocytes. Okay, so chondrocytes, these are the mature cells, chondroblasts, these are the immature cells. Next, ground substance is there. Now in ground substance, as you can see, it is made up of complex molecules containing protein and carbohydrate, and these are called as proteoglycan. Now these molecules form the meshwork that is filled by water. The carbohydrates are chemically GAG, or also called as glycosaminoglycans, and it includes the keratin sulfate, chondroitin sulfate, and hyaluronic acid. So these are the ingredients which are present in the ground substance of cartilage, which is unique for it. Okay, the core protein is aggrecan. All those things you will study in biochemistry, don't worry. Okay, the core protein is aggrecan. Now the proteoglycan molecules are tightly bound. Okay, the proteoglycan molecules are tightly bound. Along with the water content of these molecules, the firm gel-like consistency is present. Okay, so if you see the ground substance, okay, in the ground substance, all these glycosaminoglycans and this core protein aggrecan is there. Along with that, water molecules are also present. And due to the presence of that water molecule, what happens? The gel-like consistency is there, but it is firm. It is gel-like, but it is firm. Okay, that is the speciality of the cartilage. That's why it provides.
Sir, henna PVC. They are calling student section.
For what?
Tomorrow is a program for that.
Okay, so that is about the ground substance. Next, you can see fibers. So mainly two types of fibers are there: collagen fibers and elastic fibers. Okay, which are present in the cartilage. Next, perichondrium. So as I said, perichondrium is the outer covering of cartilage. Okay. Now this perichondrium is having two layers. As you can see, outer fibrous layer, which is vascular, that means blood vessels are present, and then inner chondrogenic layer, and this is cellular. Okay, cells are present, that means chondrogenic cells are present. Okay, now mostly the fibroblasts which are present, okay, immature cells, they are present in this particular layer. Now this particular layer, perichondrium, it regrow the cartilage to some extent if there is any damage. Okay, so if there is any damage, some amount of repairing can be done with the help of the chondrogenic layer which is present in the perichondrium. Next, classification of the cartilage. Okay, so three types of cartilages are there: hyaline cartilage, elastic cartilage, and fibrocartilage. Okay, so we'll see one by one. That means the important features. Okay, so please keep that in mind whenever you are discussing about the features. Though these are connective tissues, so you have to mention about the cells, you have to mention about the ground substance and the fibers also. Next, for hyaline cartilage and all the other cartilage, you have to know the location where they are situated in our body. So these points you have to note it down whenever you are studying about the cartilage. At least these points should be known to you. Okay, so first we'll start with the hyaline cartilage. Now in case of hyaline cartilage, it is glass-like transparent matrix. Okay, in hyaline cartilage, it is glass-like transparent matrix. Hence, it is called as hyaline cartilage. Now, why it is called? Because the hyaline cartilage, if you see under the microscope, mainly the matrix, it is glass-like, it is transparent. That's why it is called as hyaline. So the hyalos meaning is glass in Greek. Now histologically, it consists of cells, ground substance, fibers. Now if you do the H&E stain, the preparation matrix will appear blue, that is basophilic. However, matrix just under the perichondrium can be acidophilic. Okay, this is the difference of the matrix. That means the color of the matrix in every part will not be same. In center part it is basophilic, and just near the perichondrium it will be acidophilic. Next, the matrix glassy appearance, as I said. Now, hence the cartilage is called as hyaline cartilage. Matrix shows spaces, that is called as lacuna. Very important. Okay, so inside the matrix some spaces are there, and those spaces are called as lacuna. Now, inside the lacuna cells, these cartilage cells are present. Okay, so as I said, cells will be there, fibers will be there, ground substance will be there, right? So if you take the matrix, the spaces are there, and these spaces are called as lacunae. Inside the lacuna, cells are present, the cartilage cells. Okay. Now, hyaline cartilage matrix consists 60 to 80% of water, 15% of collagen fibers, and approximately 15% of this proteoglycans and glycoproteins. And some percentage of cells are there in the matrix of the hyaline cartilage. Next, hyaline cartilage consists of which type of collagen fibers? Type 2 collagen fibers. So as I said, collagen fibers and elastic fibers are there in cartilage. So here you can see type 2 collagen fibers are present. The fibers which are present, that is the type 2 collagen fibers. The collagen fibers cannot be seen in histological section because of the refractive indices of the collagen fibers, and the ground substance are same. Now, just now I have told you that the matrix is transparent in nature. Why it is transparent? Because the collagen fibers, the refractive index and the matrix, it is same. That's why it appears transparent in nature. Now, please find out what is refractive index. I know you have studied in physics. Right? Find out what is refractive index. So the refractive index of the fibers and the matrix are same. That's why it appears transparent in nature. Okay, because of this homogeneous glassy appearance of the cartilage, hyaline cartilage takes place. Next, ground substance, three types of proteoglycans are there. Hyaluron is there, chondroitin sulfate and keratin sulfate. Okay, these are present. Next, the proteoglycans give firmness to the cartilage. Acidic proteoglycans take violet or hematoxylin stain. Hence, the matrix of the hyaline cartilage looks violet, or you can say purple in color, which you will see in the slide. Next, the aggrecan, the core protein, which is there peripherally attached to the chondroitin and keratin sulfate. Okay, and the arrangement is in bottle brush fashion. Okay, bottle brush fashion means in the center you will be getting the aggrecan protein, and from the aggrecan protein you are getting the keratin sulfate and chondroitin sulfate, which are coming out. So if you see the molecular structure, it appears as brush which are used to clean the bottle. That's why it is called as bottle brush fashion. Next, aggrecan molecules are negatively charged, hence it is having the capacity to hold water molecules. That's why the matrix is hydrated. In the beginning, I have mentioned that around 60% water is present in the matrix. So how the water will be present? Because of this aggrecan molecule, which is negatively charged, and it can hold the water molecules. Okay. Next, cells of the cartilage. So as I said, chondrocytes are there. Now they divide and deposit in the matrix around them. The chondrocytes occupy the lacuna in the matrix. So where these chondrocytes are present, they are present in the lacuna of the matrix. Now chondrocytes lie on the side of the lacuna surrounded by empty space. Now on the preparation, okay, the shrinking of the chondrocytes takes place. That's why you can see some areas are empty inside the lacuna when the slide is prepared. But actually in the living tissue, spaces are not there. It is completely occupied by the chondrocytes. Now towards the center mass of the hyaline cartilage, the chondrocytes are large, usually present in groups, okay, 2 or more. Now the groups are formed by the division of single parent cells, and this group of cartilage cells are called as cell nest. Okay, so how the cells are arranged? First of all, cells are present inside the lacuna. Okay, now inside the lacuna, single cell is not there. 2 to 4 cells are present, and together they are forming a structure that is called as cell nest. Now here you can see the diagram. So this is the histological diagram of cartilage. This is how it will appear under microscope. Okay, when you will see under microscope ऐसे दिखाई देगा. This is how it will appear. So from here you have to identify. So first what you will identify? First you have to identify the perichondrium. In case of cartilage, always keep that in mind. First you have to identify the perichondrium. तो ये देखो यहाँ पे perichondrium है. Can you see? Okay. So in perichondrium, two layers will be there. The outer layer, that will be the fibrous layer where blood vessels are present, and inner layer will be the chondrogenic layer. Okay. Next, this particular part, which you can see over here. Okay, so see, this is the fibrous layer, this is the cellular layer. Next you can see the lacunae. Can you see these empty spaces? These empty spaces, these are nothing but the lacuna. Now inside the lacuna, chondrocyte cells are present. Okay, and you can see the chondrocyte cells inside the lacuna. You can see the nucleus also. Okay, and the appearance is called as cell nest because 2 to 4 cells are placed together. Okay, next here in the matrix, can you see the difference in the color? Some part is brighter, some part is darker. Can you see the difference? If you focus over here, see this particular area, it is more darker. See this area, this is brighter. Can you see? Now why the appearance is like this? Now 3 different types of matrix are there depending on the appearance, how they appear. Okay, so first you can see this is the capsular matrix, then this is the territorial matrix, and this is the interterritorial matrix. Okay, so once newly formed matrix, okay, the matrix which is totally newly formed, that is acidophilic, that's why the color is more dark. Once you go away, the darkness reduces. Okay, as you can see, the territorial matrix, it is less dark, and if you see the interterritorial matrix, it is mostly pale in nature. Okay, so that is the differences in the matrix, in the appearance of the matrix. Okay, next is the functions of hyaline cartilage. So what are the functions? Articular cartilage are there, provides smooth surface movement of the joint. Though it is flexible, it provides support along with the firmness. Firmness of the cartilage keeps the lumen of the trachea bronchi patent. So these are some important functions of the hyaline cartilage. Okay. Now in case of hyaline cartilage also, please note that 2 different varieties are there. One is the articular cartilage and another is the costal cartilage or costal variety. Okay. One is the articular cartilage, another one is the costal variety. Now why I am mentioning this? Because in case of articular cartilage, perichondrium is absent, and in costal variety it is same whatever we have discussed just now. So where the costal cartilage will be present? On the anterior aspect of the ribs. You have studied about ribs, right? So ribs ka anterior part में क्या रहता है? What is attached? Anterior end में costal cartilages. Okay. Okay, next is the elastic cartilage. So elastic cartilage gives resilience, pliability and elasticity to the organ wherever it is present. Now freshly dissected elastic cartilage is yellowish in color, hence it is also called as yellow cartilage, elastic cartilage. Now it possesses greater flexibility than hyaline cartilage and readily recovers shape after being deformed. So that is the speciality of the elastic cartilage. That means if you change the shape, if you make it deform, it can go back to its original position. Okay. Next location, you can see elastic cartilage is present in pinna of the external ear. Okay, then walls of the external acoustic meatus, auditory tube, epiglottis, tips of the arytenoid, corniculate cartilage, cuneiform cartilage, larynx, etc. So these are the areas, or you can say the location where this elastic cartilage is present. Okay. Now let me show you the diagram of elastic cartilage. So this is the histological image of the elastic cartilage. Okay. So in case of elastic cartilage also you can see, can you see the perichondrium? Same like hyaline cartilage, perichondrium is present. यहाँ पे भी देखो, ये देखो. This is perichondrium. So perichondrium can be seen in both the cases, hyaline also and in case of elastic cartilage also. Next, what is the difference? The difference is whenever you will see the matrix of the elastic cartilage, at that time instead of collagen fibers, what you will see? Elastic fibers. Can you see the elastic fibers? Okay, instead of collagen fibers, you will see the elastic fibers. Next point, can you see the lacuna? These large spaces, these are nothing but the lacuna. Now here the size of the lacuna are larger. In case of elastic cartilage, lacuna are present, but the size of the lacuna are larger. Next, the lacuna is covered by, or lacuna is the cartilage cells are present inside the lacuna. Okay. Now here the cartilage cells which are present, mainly the chondrocytes, these are larger as compared to the hyaline cartilage. Just see the shape of the cell. This is one cell. This is one cell. Okay. So the shape of the cells are larger, or size of the cells are larger as compared to the hyaline cartilage. Okay. So that is the important identifying feature of elastic cartilage. So in elastic cartilage and hyaline cartilage, what are the main differences? Perichondrium is present, but in the matrix, instead of collagen fiber, we are having the elastic fibers. The lacuna which are there, it is larger, and inside that, the cell which is present, that is also larger, and most of the cases, one cell is present. Previously, we have seen that cell nests are there. Okay, 2 to 4 cells are there in case of hyaline, but here, most of the cases, one cell, and in some cases, 2 cells are present inside the elastic cartilage. Okay, so these are some differences. So elastic cartilage contains ground substance, fiber cells, all those things are same. Along with that, perichondrium is also present. Next, contains dense meshwork of branching and anastomosing elastic fibers. Okay, so instead of collagen fibers, here we are getting the elastic fibers. Now these elastic fibers cannot be differentiated with H&E staining. Okay, so for that already we have discussed that what are the different stains used for different fibers. Next, elastic fibers are well demonstrated with the help of a specialized stain that is called as oracin. Oracin is a special type of stain which is used to stain the fibers. Next, ground substance similar to hyaline cartilage, but only elastic fibers are present. Next, chondrocytes are there occupying the lacuna. Cells of the elastic cartilages are bigger or larger as compared to the hyaline cartilage. Now cells are surrounded by empty spaces, it is called as lacuna due to shrinkage. So same thing, shrinking is also taking place over here whenever we are preparing the slide. Now chondrocytes lie singly or in group of 2, not more than that. Means cell nest appearance is absent over here. Okay, now this you can see the areas where different cartilages are present. So hyaline cartilage, where it is present, epiphyseal plate, ribs, respiratory tract, articular cartilage, etc. These are the examples of hyaline cartilage. Next, elastic cartilage, you can see these are the examples. Okay, so whenever, see, when you were child, I don't know all those things happened with you or not. When you were child, at that time, suppose you are not listening to your parents or you are not listening to your teacher, what they used to do? They used to pull your hair, right? They used to pull your hair like this. Then when they will leave it, it will go back to its original position without any deformity. That cannot be done with bones, right? Why? Because of the elastic cartilage. Okay, so please remember that whenever elastic cartilage, pull your own hair. No, this is the shortcut. Don't laugh. During the time of exam, this will help you. Now you are laughing. This is the time of exam. This will help you. जब भी elastic cartilage आएगा, क्या करना है? कान पकड़ना है, ऐसे करके. Then you will remember that यहाँ पे रहता है elastic cartilage. Okay. Next, fibrocartilage, intervertebral disc. Have you heard about intervertebral disc? Okay, so in intervertebral disc, this fibrocartilage is present. Okay, next, the last type is the white fibrocartilage. So perichondrium is absent. So see, this is the most important part of the white fibrocartilage. That is, perichondrium is absent. Okay, now, and it is having thick bundles of collagen fiber, that also type 1 collagen fiber. Previously, in case of hyaline cartilage, we have seen type 2 collagen fibers are there, but here type 1 collagen fibers are present. Okay. Dense regular connective tissue like appearance can be seen. Minimal amount of ground substance and few cells are present. Why? Because mostly it is occupied by bundles of collagen fibers. That also type 1. So those bundles of collagen fibers are occupying most of the ground substance. So that's why there are very small space left, and in that space only the ground substance and cells are present. So mostly it is occupied by bundles of collagen fibers. Okay, that's why the appearance is white in nature. Now chondrocytes are seen between the fibers, that also they are placed in rows. How the suppose one bundle, another bundle, in between these two bundles the cells are placed in rows. Okay, so I hope you can see the diagram. In this diagram, as you can see, perichondrium is absent. Okay, perichondrium nahi rehta, perichondrium is absent. Next, here you can see the bundles of the collagen fiber. These are the bundles of the collagen fibers. Now, in between the bundles of the collagen fiber, can you see small spaces? In those spaces only the ground substance or the matrix is present. And just see how the cells are oriented in rows. Okay. So one bundle, another bundle, in between them the space which is there in that rows are there where these chondrocyte cells are present. Okay, that is the structure of fibrocartilage. Okay. Now what is the function of fibrocartilage? Fibrocartilage provides support and rigidity. Okay, it will provide support as well as rigidity to the surrounding structure, and it is the strongest of all the types of cartilages because it is having thick bundles of collagen fibers. Okay, now what is this? This is the gist of the story. Whatever we have discussed till now, okay, this is over here. So here you can see hyaline cartilage, elastic cartilage and fibrocartilage. Okay. These are the charts will help you to revise entire chapter in 5 minutes. That's why every day I hope you have observed that after completion of one chapter, I used to make a table and then I used to show it to the student. Okay, so entire chapter, whatever we have discussed, the gist is over here. It will help you to understand about that particular topic. So first you can see extracellular matrix, cells, the arrangement of the cell, perichondrium, whether it is present or absent, the location and the function. So all those things only we have discussed, but here it is given systematically. So just see in extracellular matrix what is present over here and over here. Okay. Now this you have to study from your own. Whatever we have discussed, whatever you have understood, from that you have to study from your own and then just compare so that you will understand about the cartilage, whatever we have discussed. Okay. So you can see the extracellular matrix what is present in all three cases. Next you can see the cells. Then you can see the arrangement of the cells, how the cells are arranged. Next you can see the perichondrium, where it is present, where it is absent. Then you can see the location. This part is very important. Okay. See, these are the locations of hyaline, these are the locations of elastic cartilage, and these are the locations of fibrocartilage. Okay. So along with the intervertebral disc, pubic symphysis, menisci, glenohumeral, labrum, okay, temporomandibular joint, these are the areas where fibrocartilage is present. Next function, you can see joint articulation, scaffold for osteogenesis, means when the formation of the bone is taking place, at that time it is helpful. Then it provides structural support. This one weight bearing, mainly the weight bearing parts of our body. For example, vertebral column, it is also bearing the weight. So in that area, this cartilage or fibrocartilage is present because this is very strong. Okay, then it can resist the compression force and then tenacity. Okay, tenacity means mechanical force, where compression force, mechanical forces are applied in that particular area. This is present. Okay. So that is all about the cartilage. So today we have discussed what is connective tissue, what are the components of the connective tissue, right? In components, what are the different types of cells, what are the different types of fibers, and then about the ground substance, right? Then we have discussed about the classification of connective tissue, where we have discussed generalized connective tissue and specialized connective tissue. Generalized, all we have studied along with the examples, and then in specialized connective tissue we have studied about the cartilage. In cartilage we have seen three different types of cartilages. Okay, hyaline cartilage, elastic cartilage and fibrocartilage. Important identifying features of those cartilages and how to identify under microscope. Okay, then what are the functions of those cartilages and what is the location. Okay, that much we have studied today. Okay, now what we will do, as I said, you have to identify. Okay, the distinct layers of the perichondrium is visible. Yes. Now focus on the brown substance.
Yes, sir.
Yes. Can you see the lacuna?
Yes, sir.
Yes. Now inside that, I hope you can see the chondrocytes.
Yes.
Yes. Yes, chondrocytes are visible. Okay. Now try to identify the cell nest structure. Just now we have discussed. Try to identify the cell nest structure. That means inside the chondrocyte, inside the lacuna, you will be able to see the cells. Multiple chondrocytes are present in a group of 2 or 4. That is the cell nest. লেকিউনাটা আছে, লেকিউনাটার ভিতরে ইনসাইড দ্য লেকিউনা, ওকে, যে কন্ট্রোসাইটসগুলো আছে, মাল্টিপল কন্ট্রোসাইটস আছে, ঠিক আছে, দ্যাট ইজ দ্য সেল নেস্ট থিংস। Okay, so during the time of exam, ऐसे रहेगा microscopes, ठीक है, like this microscopes will be placed, okay, and this particular area it will be covered. So you have to see within 2 minutes, you have to identify, you have to write the identifying feature, and then you have to go to the next slide. Okay, for that you have to practice. Whenever you are getting free time, please come and practice. This is very important. Save it. Okay, and whenever you are free, see the images and compare with the identifying features which they have given. This visualization is very important. Okay, to see, visualize, try to identify the important features of the tissue. Okay. So today we are having slides of hyaline cartilage and elastic cartilage. Fibro is not there. So for fibro, the diagram which I have shared, so you have to take the help of the diagram. Remaining both the tissues, I will show you for the slide. Yeah, pericardium is present in both the cases. What else you can see? Yeah, lacuna is there. But here in this case, lacuna are larger. Please focus on the lacuna. Okay, and then you will be able to see only one chondrocyte inside the lacuna. Okay. Next, in between the lacuna, you can see this wavy type fibers. These are nothing but the elastic fibers. I hope it is visible. Okay. So this is how you have to identify the elastic cartilage. In elastic cartilage and hyaline cartilage, the difference is first focus on the lacuna. In the lacuna, you will see it is larger as compared to the hyaline cartilage. Okay, and then you can see the branching pattern of the elastic fibers. From there, you have to differentiate. Okay, next. Okay, next is